and some of the classification of the immunoglobulins, and they are classified on the basis of their heavy chains. According to the type of heavy chains, they are divided into 5 classes. Mainly the immunoglobulin classes are the A, IgA, IgG, IgD, IgE and IgE. Here the IgA has the type of heavy chain as the alpha, and the type of light chain can be either the kappa or a lambda. This is happen to all the immunoglobulin. They can be light chain, can be a kappa or a lambda, but only one, not the both. Okay. Then the gamma chain is present in immunoglobulin G, and the delta is present on immunoglobulin D, epsilon immunoglobulin E, and the IgM has in the mu. Then there is also a subclasses such as IgA is divided into 2 subclasses, IgA1 and IgA2, and IgG has the 4 subclasses, IgG1, IgG2, IgG3, IgG4. And the IgG, this is the most abundant immunoglobulin in the plasma, and they're having the longest half-life and performs all the effector functions and can cross the placental barrier also. And these having the maternal IgG provides the immunity to the newborn in the first week of the life. So IgG is very crucial, and if the IgG is lacking, the newborns are also lacking with these types of IgG. These are the structure of the IgG. Then the IgA. IgA is present in the plasma as well as in the exocrine secretion, and the these exocrine secretion is known as the secretory IgA, and the plasma IgA is the major is a monomer, while the secretory IgA is a dimer. This is the IgA monomer, and this is the secretory IgA. This is in the dimer form, joining through the J chain. These are the secretory components, and this is J chain, and these are the IgA monomers, and this is IgA dimers. Then the secretory IgA. This secretory IgA is formed beneath the basolateral surface of epithelial cells, and it binds to a Ig receptor present on the epithelial cells. A part of the Ig receptor detaches from the cells, and it attaches the IgA dimer as the secretory component. Here we can see with the diagram, this is the plasma cells, and after that the plasma cells, and there is the synthesis of the IgA. These IgA are then dimerized, and they react to the IgA receptors, and from there they are engulfed out, and they are transported through the vesicles and then released out in the lumen, where they interact with all the antigens. Then the secretory IgA traverses the cell to reach the mucosal surface, and it acts mainly by the neutralization process. And the secretory IgA present in the breast milk enter the gut of the newborns, and it binds to the mucosa and protects the baby against the gastrointestinal pathogens. Now the another class of the immunoglobulin is the IgM. This is the largest immunoglobulin, and the first immunoglobulin to be secreted upon entry of any antigen, and this is the only first response towards the entry of any antigens, and acts mainly by the complement activation, and this having the weak neutralizing and the opsonizing activity, but they are very much important. They having the pentavalent structure. These are the structure, and they are mainly formed in 2 forms. This is the monomeric form, and these monomeric forms combined in the group of the 5 pentameric form, and they form the J chain. using the J chain, and this forms the IgM. Then the IgE. IgE is bound to the mast cells and the basophils, and these parasitic antigens bind to the IgE on the mast cells. And mast cells, what they do, they release the chemical mediators such as histamines, and they can destroy the parasite and also mediate the allergic reaction. So IgE is very crucial, and these if the IgE level increases, that means there is the any kind of the allergic reaction taking place inside the body. This is the structure of IgE. Then the IgD, this is very less known immunoglobulin, and the function is not known properly for this immunoglobulin D. Then the important feature of the immunoglobulin classes here, that in the table we can see different classes of immunoglobulin and their concentration in the plasma. Here the immunoglobulin G having the highest concentration, whereas the D having the least concentration. As compared in the half-life, where the IgG having the half-life of 23 days, is that is the highest, whereas the other Ig AME having the only the 5 to 6 or 2 to 3 days of the half-life. And what they do, the basic things are the IgG, they are active against the bacteria and viruses. IgA is strongly antiviral and weak antibacterial, whereas the IgM, this is the first that is encountering with any kind of antigen, they are strongly antibacterial, whereas the weakly antiviral, whereas the IgE, they are the antiparasitic mediators and the allergic reaction mediators. IgD and its function is lessly known up.